everyone, for the second talk of the day. It's my pleasure to introduce Bianca. I know her for a while and she's fantastic. She did her PhD at Princeton under the supervision of Barbara, and I'm very bad at saying her name. Can you do it? So
Engelhardt,
thank you. Then she was a lecturer in Cambridge, and now she is an assistant professor at Columbia, jointly between the Department of Statistics and Computer Science, right? And also I think the Ireland Institute of Cancer Dynamics. She does very interesting work in the space of computational biology. And so please join me in welcoming her.
Thanks, Morgan, for the really wonderful introduction. And thank you all for being here on a Saturday in this basement when the weather is so nice outside. So hopefully this will be exciting a little bit. I'm very happy to give this talk to such a diverse audience. I think I've been learning a lot throughout the session, both yesterday and today, and I see a lot of very interesting overlaps even across fields. So if you have a lot of questions, you feel free to ask during the talk, but I'll also be available throughout the day. So if something inspires you or you feel like you would like to maybe collaborate or think about some problems together, I'm always excited about that. And today I'm going to talk to you about some new work that's coming out from my lab. We're the computational morphogenomics lab, and that's a bit of a mouthful. Morpho means I'm very interested in how things look like, so in their morphology, and how they interact with the genomics part, so what's inside cells. The driving questions in our group have to do with what makes up cellular identity. So we're made up of many, many cells, you know, we have lungs, eyes, all sorts of things that allow us to be functional. And these are made up by hundreds and thousands of different cell types that are working together in non-trivial ways to give rise to a lot of diverse functions. So I'm very interested in how cells interact with one another to generate shape and form. And the long-term goals of our lab is to kind of develop digital twins in a cross-species comparison way that allows us to uncover principles of development. So what that means is that there's a lot of really cool species out there that, for example, can regenerate. And I'm very interested in learning from them in order to heal more efficiently. And I like to do that by developing mathematical models to collaborate with my wet lab colleagues that can actually collect the full data to get us there. So we live in a world of generative AI, and a lot of the tools you'll be hearing today belong to that space. From more classical statistics to transformers, but it's an exciting time for biology, but it's also kind of a time for cautious optimism in the sense that as these tools become more and more complex, it's very important to understand the underlying statistical assumptions that can lead to particular predictions. So today we're going to look at one particular type of organism, and that's the mouse. And there's many, many different ways of looking at mice, and here are just a few of these. It's a very exciting time to be a computational biologist because there's a lot of very different data sources. So what you're looking at right now to your left, top left, is a live sheet microscopy recording of mouse development from Kate McDowell's lab. It's very exciting because if you zoom in, actually, you can see cells at individual granularity. You can see them building up vertebra in this very early mouse. And that's a phenomenon that's super fascinating. There's a lot of movement, there's a lot of pushing. So there's a really, it's an active material that's becoming more and more functional as time goes by. At the bottom, you can see later stages of development where you can actually see the morphology of the actual mouse taking shapes. And in the space of computational biology, we're actually looking at what's happening inside the individual cells. So this is very much on the right part. And a lot of the tools that have to do with, I'll go over the types of data, that we have available from these particular experiments, and I'll tell you a little bit about the computational ways of relating these data sets in a little bit. But so what was very exciting today is that we actually really got to hear a lot about electronic healthcare records and about kind of how do we use big data to study disease. What I am primarily looking at, I'm trying to understand a little bit how does the cell and what the cells contain can be used to understand how a functional organism is formed. So we heard a lot about this part of this paradigm, and I'm here to kind of take it back to the basic science a little bit. As I said, it's exciting to be a computational biologist because we're benefiting from data from many sources. So in the same way you have data from multiple hospitals, we also have data from multiple consortiums. And of course, the noise is going to be different from whether you have patient data from multiple hospitals, but there's the same issues of variability and variance and parameter identification raised because all these consortiums aggregate data from multiple labs. Everyone is working at different times of the day. The protocols try to be the same, but there's always some human aspect or processing that can get involved in the preparation of samples and so on. So it's very exciting, but there's also a lot of opportunities. And in particular, in the past few years, there's been quite an explosion in the type of literature that's been put out on this topic. And I really enjoy Neil's talk yesterday when he was telling us about, like, what are the topics that are really emerging? How do we navigate this explosive literature when we want to make progress? So for like, one of the things that I wanted to emphasize is that, okay, we have a lot of data, there's a lot of opportunities. What are really the challenges that us as computational scientists or statisticians can address? And these generally fall, I'm sure I missed some, but in my mind, a lot of these kind of like span the 6 thought bubbles, and they have to do with, like, how do we model noise that comes from multiple sources? How do we find efficient representations and dimensionality reduction techniques to represent this data more efficiently? Very excited about Melanie's talk after this because she's probably going to talk more about the geometry, and I'm very excited to learn more in this context. Another very important pillar is that of interpretability. So we do have all these sophisticated models like foundation models and large language models, but what do they actually teach us? Can we extract scientific insights that allow us to perform experiments beyond saying, here's this cocktail of drugs that I don't know much about, how will that impact the effect of a patient? Interpretability and mechanism are very related. By mechanism, I mean, how do we incorporate information from biophysics? Remember, I was telling you embryos are active materials that are intelligent, they're adaptive. So a lot of the research has been treating these organisms in a way in which it kind of dissociates them into multiple cells that then don't have much feedback mechanism. Well, that's not really the case. So there's a lot of, I hope to convince you in the talk that there are some lessons from biophysics that as geneticists we can incorporate and kind of try to learn from or influence. The last bubbles, I'm not going to be talking too much today, but they are very important. One of them is experimental design. As data is becoming larger and we can intervene in more complex ways, how do we choose the experiments we're doing in an intelligent manner? So for example, as the space of design is combinatorially complex, how do we use time? And we need people, we need graduate students, researchers to do all these experiments. How do we prioritize that needs to get done in the lab? So having a lab in the loop kind of formalism for this problem is very important. And last but not least, scalability. As this data becomes very large, we need to store it, we need to load it, and we need to make sure that we can actually convert it into educational tools for new generations of scientists to learn what are the best data processing pipelines and frameworks for trying to learn about this data in the space of thousands and thousands of methods that are coming up every year. So I'll be talking mostly about these particular pillars today. And before I get started, I know a lot of you, most of you actually might not be, maybe we can do a bit of an acrobatic exercise. Don't worry, I'm not going to make you do handstands, but can I have raised hands if you work with gene expression or you have a working knowledge of gene expression in chromatin accessibility? Okay, so a very small number of you, which makes me excited because then I don't have to pass through this slide very quick. Let me tell you just a little bit about what is there in computational biology, like what are the tools that we're actually working with. So there's a lot of modalities. People usually use the word multimodal in computer science to talk about text data or image data. In biology, it really refers to the type of information we can extract from the cell. So there's maybe you've heard about the central dogma. Central dogma is a kind of long-standing theory in the biological field where we say like, okay, you know, each cell contains DNA. DNA is then used to make mRNA, and this mRNA is used to make the proteins that are the functional modules in your bodies. And each cell uses this DNA information in different ways in order to accomplish functions that are essential to the organism. When we talk about gene expression, we mostly talk about the quantity of mRNA that corresponds to a particular gene. So if you're to, you know, I'm sure maybe a lot of you use Netflix or Hulu or any sort of streaming platform or Twitch if you are a gamer. But imagine this is like, you know, this is a movie, each cell is a movie, the genes are the features. And basically, you know, this is kind of like a Poisson count matrix. Each entry here can be either 0 or usually positive. It's an integer number that tells you how much of that gene is present in your cell. And it's been quite successful at helping us identify different cellular compositions. But in addition to these numbers, there's all sorts of other other quantities. So for example, the DNA can be annotated in different ways. Imagine that you have, like, if you're familiar with the Turing machine from computability, you have this like 001s where you're marking the tape. That's kind of like, there's an equivalent to that in chromatin accessibility. There's a lot of information about the spatial coordinates, so where the cells are in space and time. There's information about how the cells interact with one another. There's information about what the cells show on their surface in order to detect their neighborhoods. So there's a lot of layer of information that creates a lot of opportunities for a lot of methods out there. And in this, I really like this review from Sarah Teichmann's group, where the authors are trying to kind of create a compendium and what are the different types of modalities that exist in this space, from DNA to chromatin accessibility, gene expression, and then protein abundance, and trying to understand, okay, like, what are the key tools that we're using as a field? And most of the time, these tools have to do with three main components. One of them is transfer learning. We listened to the wonderful talk earlier about how to make predictions in unseen conditions. Another tool is that of matrix factorization. We usually have some big matrix that we want to decompose as the product of components with features and how important those features are to the context. And the other one is really old school linear regression model. And, you know, if you become more fancy, you can do a lot of deep learning on top of that. But these are kind of like the bread and butter type of tools that computational biologists use and reuse in different contexts. And these tools, while simple, become difficult to apply when the data becomes more complex. So for example, when I'm talking about scales, let's say I'm talking about images, well, images can be quite diverse. Maybe I'm talking about vasculature or a network of that looks kind of like a tree, or maybe I'm talking about individual cells that can be tagged. When we say images, we can maybe look at the number of genes, like each dot here represents a particular protein or like molecule that's present in the cell at a given time. So it's much higher level resolution here, but maybe I don't know where the boundary of the individual cells are. So all this information is complementary, but it tells a very different story in the right biological context. So understanding what's the right scale and how to quantify it is something that's quite difficult, and what makes progress, synchronized progress in the field, quite challenging. Another important scale is that of how the points are organized in space. So if you remember Rashchetti's talk yesterday, you saw a lot of these really interesting neighborhood maps where you would see, for example, the map of Manhattan with different areas partitioned based on their zip codes or other features. In the recent 5 years, with the advent of spatial transcriptomics, we have the ability to look at where cells, what is the place that they occupy in space. So that's been allowing us to make the same maps that we make for geostatistics for biology. And this has been really exciting because now we can ask, okay, not only what are the different cell types, but we can also ask what are the patterns that are collocated with particular organs or particular stages of development. So that's been really powerful. And the main tool that's been used is that of Gaussian processes, because this allows us to kind of look at correlations that are both short and long scale and be quite flexible in the choice of kernels. And I'm highlighting Gaussian processes here because this has been also a tool that's been very popular in the context of electronic healthcare records. So if you have data from patients that have a bunch of covariates, like, level of sugar, you have a lot of measurements, Gaussian processes have been used quite a lot in interpolating data that's not aligned. So two different fields, the same tool, different opportunities for modeling. So how do you aggregate all in this information across scales? Well, one of the main things that we're interested in our lab is quantifying the morphology and relating the morphology to tools like, kind of clustering, representation learning, in order to associate the morphology with another modality of interest. And one of the main tools we've been developing and building on for some time now has to do with this idea that, okay, like maybe there's a smart way in which I can use all this different data, I can process it, and I can project it into some space that retains some original properties of my information. So let me be a bit more specific about. So let's say I have a set of objects on the left. This stay in space of dimension d1. And let's say I have a different set of objects that stay in some space of dimension t2. And I would love ideally to put this object in some space where I can learn a representation of the matching of the corresponding object. So let's say I have a cell and I can collect morphology on one end and I collect gene expression on one end. And I would like, how do I represent these two different types of information such that I can cluster them together, or I can say what corresponds to what. And this is really nice, and people use it a lot for visualization. What you typically do, you take this joint object and then you project it into a space, like a 2D space or 3D space. There's a lot of distortion that happens, but you use that space to visualize the data at hand. However, this particular model doesn't allow you to answer questions that would be very interesting in practice, such as if you only have access to some modalities for a study and not the other one, how would you be able to recover one from another? And the other main issue that it doesn't take into account is that, like maybe not everything in the two modalities can be matched together. So maybe there are some particular aspects of your space that are only specific to a modality and not the other one. So how can we learn? How can we interpret this latent space? And how can we use this latent space to recover the things that we haven't seen? So how does that look like mathematically? Well, this looks like a multi-study factor model where there is a latent variable z. I made this particular figure for two modalities, but you can have 3 or 4 or as many as you want. You can imagine that the two main components here is how do you represent the data? So like how do you represent morphology and put it in a space that's regularized. So this is each zk corresponds to features that are only specific to one of the modalities, but each modality is decomposed into something that's common and something that's study specific. So this is a generalization of a canonical correlation analysis method. It's a probabilistic version of a very classical algorithm due to Hotelling from 1906. And mainly the challenges that present themselves in making these models useful to biology is parameterizing this function f in the right k and adding the right sparsity structure on A and B such that these components are identifiable. So those are the main computational statistical challenges that arise. And of course, interpreting the results together with the biologists. So in one application for some time ago, we looked at how these models can be used for extracting information from histology slides across multiple tissues and trying to understand what is the correlation between the underlying bulk gene expression information from multiple patients. And we try to ask the question of whether, if I only have the gene expression, can I predict what would be the fantasy slide, kind of the digital twin replica of this slide using our joint mapping. But so this type of approaches can give you a couple of things that are exciting. So you can represent cells in a composite way. You have multiple modalities, you project them in a space. You can then play with the space to say how informative is this for variation. You can integrate spatial and temporal data because there's no reason why you could not turn this model into a car-like model, like conditional autoregressive model or so on. You can add all sorts of noise that incorporates spatial or temporal correlations. So these models are very, very flexible. And you can also recover one modality from another. So if you have missing data where it's very difficult to collect everything at the same time, you can make use of such models to solve that problem. However, there's also something deeply unsatisfying about this because you take all this very complicated data, but there's also a lot of information about the structure of an image that we know or people from other fields know, like in biophysics. So it's nice to go to some neural network, but maybe there are situations where we could use information that already exists to kind of uncover actual knowledge. So I'm very interested in how do we do that in situations where the problems are a bit more precise from a biological point of view. So for that in particular, one of the questions that I'm very, very obsessed with these days is, okay, so tissues are shaped by mechanical forces. It's kind of like this 3D origami setting. And where do these forces come from? So, like at the beginning, you have kind of this blob of cells that are kind of pushing against each other and moving around. Where are these forces generated? So, of course, cells produce all sorts of components. What of those components are actually most correlated with forces you can observe in a tissue slide? And this problem has a long history in the biology and biophysics literature. Eric Wischhaus, in particular, has spent a lot of time in the context of flies to think about these actin-myosin networks that he proved have a lot to do with how changes in tension between cells is happening over time. But a lot of this work is very hands-on in the sense that there's some genes or proteins people know about in the field, and they do a handful of experiments. So could we use tools from genomics and statistics to do a de novo discovery of new genes that maybe we can experiment on? Maybe we're missing, I didn't tell you about the scale, but in a mouse or human, you have around 40,000 genes. Maybe out of these 40,000 genes, there might be a lot of them that could have interesting mechanical roles, but we just don't look at because historically, those have not been the ones that have been most popular. So we set to do something quite ambitious, and what that was to quantify how the multiple modalities, one coming from morphology and one for genomic, interact to tell us something about space and about boundary formation. What do I mean by boundary formation? So when you start forming an embryo, all the cells look more or less alike. But at some point, and that happens quite early on, cells begin to separate. Something becomes a heart, something becomes a lung. At which point does the separation happen? How do the cells know how to choose their own fate? And how do they do this in such a synchronized way that they know how to not move across domains? So this is a really, really fundamental question to which we don't have an answer. I mean, there are case studies here and there, and there's a lot of knowledge, of course, but we do not have a clear understanding of whether that's because tension creates barriers that forces cells to not move from one part to another, or genes are creating barriers of expression that don't allow particular tension profiles to form. So there's a little bit of a chicken and egg problem that we would like to solve. So we would like to understand what are the genes that are impacting tension and what are the genes that are responsive to tension. And we would like to find, in an unbiased way, a pretty large set of genes that would have this behavior. And we would like to do so such that then we can go back in the lab and do experiments and see whether we're actually right. So we're not yet at the part where we're going back to the lab to do these experiments, but this is about the statistical study on how you can begin to come up with candidate genes for doing that. So there's a couple of, this is work done with my fantastic master's student at Cambridge, now my PhD student at Columbia, Young, and with that biophysics collaborator, Adrian, who helped us a lot with the simulations that I'll be showing you in a moment. We looked at the mouse embryo across multiple embryos, particularly at the midbrain, hindbrain area, because at this particular stage of time where we had, this is E8.5, there's around 30,000 cells in the embryo at that moment. And it's a very crucial moment for forming the neural tube and a lot of neuronal subtypes that will make up the brain. So there's a time for a lot of movement, but also a lot of separation and boundary formation, which is why we chose to look at this. And we used two particular ingredients that were exciting. One of them is this really wonderful neural network for segmentation. It's been around for quite a few years, and it's been really helpful in a lot of contexts where you're presented with data that looks like this, and you want to find out where the borders of the cells lie. So unfortunately, you cannot use it out of the box. You always have to do some modification. You need to do some manual annotation. Your curation has to be really precise. So actually, one area of active research is how do you minimize this need for manual curation as you get different types of images? Because as the microscope gets better, these images kind of get a little bit different. So you have a lot of different technical noise. And every single time you have a different problem, you kind of need a graduate student of a postdoc to do a few more annotations. And that's really inconvenient. I mean, it's doable, but we would like, with the advent of such nice technologies in machine learning, to solve this problem, to not have to worry about it. But this was sufficient for us. So we did a modified unit neural network for segmentation, so we can extract the boundaries of the cells in our study. And the other ingredient that was quite exciting is that we found out that there actually exists a lot of work in the biophysics literature about how, in soap bubbles in particular, we found that there's ways in which if you have an image of cells, and this is segmented in a way that you have multiple polygons and vertices, what you can do, you can actually fit tension profiles that you are solving for with high school physics. So that was pretty exciting. You do a first balance analysis at points of intersections between the cells, and you can actually come up with some estimates of how much tension does a cell feel across its boundaries. And there have been a number of studies that have correlated these measurements with real tension measurements across a number range of organisms. So this is not just something that you can do in a particular toy case example, and it doesn't carry to the real biology. But this was very exciting to us because what we thought about was that, okay, we have these two different tools. What would it mean to think about them jointly? So we put together this pipeline for joint tension and genomics exploration, where we refined a lot of the segmentations required for this data, and we did refine the process for extracting this tension inference from images, together with a few statistical models that I'll show them to you in a moment, in order to understand, okay, how are these tension-related phenotypes organized spatially, and how are they correlated with differences across boundaries and gene expression. And we were very surprised to find a number of really exciting things. And some of these things were the following. Across boundaries, you can actually see that if you were to estimate pressure and stress tensors in the slice, you could see some sort of a decrease from the boundary and away from the boundary, which is completely expected, but this was more kind of like a quality control. Does this make sense? Will applying this tool give us something that's replicable? The other interesting thing was that if you took slices horizontally, there was still quite a lot, because you can imagine that when you have these slices, you might capture multiple cells. So you're not going to capture only one cell in one slice. You might have like different, and maybe 2, 3, 4 in one slice. So there's no reason why the tension should be exactly the same. But we actually found that in the area closest to the boundary, we had, again, like very high correlation in terms of this slice. So it means that there is some signal that's traveling throughout the tissue that kind of informs the cells of what the actual tension is. So how do we combine this with the genomics information? Well, we leverage these particular models from the geoadditive structural analysis community because remember, our question was, and we do have a lot of causality experts here, so if you want to talk to me about this after this, I would love your opinion. But basically what we wanted to do, I told you at the beginning, we're interested in finding genes that are responding to tension and genes that could maybe affect tension. But one of the things that we did in order to do this, we used space and force balance analysis to get the tension. So space kind of indirectly affects both of these two measurements. So if I were to just like fit in a Gaussian model, just a linear regression model to find out what my contribution would be here, that would not be really great, because maybe I will pick up a lot of genes that are just purely correlated with space. And this is something that happens quite a lot in biology. You do have what are called morphogenic gradients that are either from the mother or that are getting created in the process of development. So we don't want to find out those as important. So we did a simple spline model that regressed out the spatial location. And then instead of solving a linear regression model for the association between gene expression and tension, we simply looked at the residual contribution, building on some really nice work in this space. And of course, there's a lot of choices to make, like how do you choose the right parameters from the spline model? We had to do a lot of robustness checks. But one of the things that was really fascinating is that we actually uncovered mechanosensors that were identified by a biologist over the past 20 years. So at the top of our list, we also found, and interestingly, we also found groups of genes that people haven't characterized before. So it'll be really interesting to look at. So in some sense, this gave us confidence that this tool is good at picking up very strong signals that people have validated in the past, but there also could be additional genes that can give us more insight and more nuance into understanding this process. So a lot of these, for example, are they're called ephrins, and you can see very clearly that a lot of them have really strong patterns that are separating these boundaries. And again, like, I'm very thankful to be at this workshop, because when I was looking at the talk yesterday on economic mobility, a lot of the tools in that space are very much related to this idea of like, how do you detect the boundaries? How do you understand whether the mobility of a cell, impacts what it will become? Like, I think a lot of these questions, even though they sound philosophical, they could occupy very similar space. So I'm very excited to revisit that literature starting tomorrow. But you can see like there's a lot of these clear boundaries, and here this boundary is based on expression. But you can see, I think here it's a little harder to see, but you can see that there's also tension across the boundary that separates this particular gene expression. I mentioned to you some simulation. One of the things that was what we did was we asked also the following question. We know in development that the boundaries that so we wanted to ask, okay, are all these boundaries created equal? So in other words, once you have a boundary that's based on gene expression, when will those cells mix and when will they stay apart? So from the theory of this nice biophysical model, you can say that there's like some particular rule across the boundary where the tension at the boundary is higher than the average of the tensions to the left and to the right. So there are some specific conditions under which if you do a simulation, these two cells will not, sets of cells will not mix. And what we found very interesting is that we did find the same signature in our real data. So basically, we found boundaries of gene expression where the cells kind of stopped mixing and the two parts are forming different areas of the body independently. And we also found kind of like boundaries that were associated with a lot of mixing, where like the flow of cell was not done yet. So we did some cellular thoughts model simulation to kind of see, okay, like what happens if you have different initiations of your tension profiles. And we're exploring this further, but essentially what happens here is that with a lot of the boundaries that are very well established, you actually predict that there will be no flow between the cells going left and right, which agrees with the biology. That's very exciting to me. So what did we learn by integrating these two? We were able to recover known mechanosensic genes, but we were also able to identify new genes that we are going to be exploring experimentally in optogenetic experiments. We were able to characterize boundaries based on these two different types of data. And what's very interesting is, well, it's very good we can do this in 2D, but biology doesn't work in 2D. So what we ideally want to do, we would like to build towards 3D models. So we already know that tensional profiles are very important in the quality of organoid assemblies. So one of the directions we're exploring is like, how do we take these 2D tools for estimating tension to a 3D context where we can actually look at cells as the real volumetric objects that they are. And it was like, we were very excited. We're like, okay, this is what we're going to do next. But then we realized that there's actually very little in terms of, if I give you a lot of high dimensional objects that are this tiny spheres moving into 3D space, there's very little work on how do you aggregate this information from multiple replicates. So in other words, if you have a fly embryo, easy to aggregate because it basically does the same thing. Like it's very, very similar, especially in the early stage of development. But if you look at something like mouse or maybe human, there's a lot of variability of the paths that the cells are taking. So it's very hard if I give you a couple of videos frame by frame by this large light sheet microscopy recordings, it's very hard to say, what would this cell here correspond to in here? And why would you like to do that? Well, as statisticians, we want to talk about timing. We want to talk about replicability. We want to talk about noise. Noise is very important in biology. Whether some phenomenon is more precise than other can tell us quite a big deal on how many resources are being allocated there. So it's very important to solve this engineering project before we even embark on integrating the live imaging with the spatial transcriptomics that I was mentioning to you. So again, the goal is to understand this notion of memory, through attentional memory from space and time. So far, I didn't have access to time shots. I could only characterize the relationship between genes and tension. But what I'm really interested in understanding, how is the cell type affected by a sequence of tensional changes? So in order to do this, we started to put together a lot of groundwork using tools from transformer and vision transformer literature. And we decided to start thinking about what is the best method to take multiple embryo recordings and kind of put them into the same video, into the same common time frame. So in order to do this, we, together with a very talented postdoc in my lab and in John Marioni's lab at Cambridge, John Marioni is now at Genentech, but we did quite a lot of work in trying to understand what is the best machine learning tool that would allow us to solve the following size. So given that I give you a query slice from this image, this is kind of how it looks like. Could you tell me what would be a corresponding query size that recaptures the same biology in another embryo? And this problem, when we first started of it, I was like, oh, it's probably already solved, because can you just compute the distance between all the pairs of the of such images, and then can't you just do some like maybe optimal transport or something that already exists to match them? And what we learned actually for the type of data in light sheet microscopy, the way you compute distance and similarity between consecutive images is quite complicated. Why? I would love to know. But so far we found that a lot of the existing tools really fail at detecting the right correspondences. So we went on to build our own framework, and we actually found, so we found out that the best solution for solving this problem has two main ingredients. One of them is using shifted window transformers, which are a class of vision transformers that instead of predicting the next kind of word, they predict the next patch in an image. So it's kind of like generalizing. We did not come up with it. It's a technique that basically takes the attention mechanisms and apply them to images rather than text. And we also found out that actually just by using this by itself is not quite good. So one of the things that we need to do is also use another tool, which is called contrastive learning, which means I'm helping the algorithm by giving it some positive samples. So instead of telling it, hey, here's my data projected into a space, I'm going to give my data in pairs. So I'm going to say, okay, like some of these pairs are very similar. I can give it similar pairs because I have access to consecutive time windows. And I can also give it very different pairs where I can maybe match one slide in one embryo to a very late slide in another embryo. And that would be an example of a negative sample. And we found that this actually improves our problem quite a lot. We're still refining this, but hopefully we'll have a preprint quite soon. So I'm very excited about this direction because once we have this, we'll be able to layer in the transcriptomics data, and we would also be able to do the segmentation of the 3D volumes and ask how are different cells different across embryos. We can quantify all sorts of variance and timing and interesting phenomena. So that should be really, really neat, and I'm very excited about it. Getting ready to wrap up. I've been talking to you about how do you represent these developmental scales in genomics. But what is even more interesting than visualizing data and representing it is actually learning rules from it. So I don't have I wanted to play it safe, so I have a PDF of this, but there's this really cute paper here on, okay, so has anyone here played Minecraft? Maybe it's not the right audience, but any Minecraft player here? A few of you, huh? I'm happy. I'll take what I get. You know, there's 4 of you. How many played Hollow Knight before? Someone, nobody? No? Okay, so my character, somebody that recognized a little character. Hopefully they don't sue me. Anyway, so it's just free advertising. It's a great game, guys. So basically, what is Minecraft? Minecraft is a game that's very popular in which people can construct from cubes various structures or whatever they want. And it's kind of interesting, you know, kids go there and they can build a whole Columbia University campus. You know, there's some labs that actually use it. A colleague of mine has a virtual lab on the Minecraft server where he has like chickens that he sequences. They're all virtual. It's kind of fun. But the point is that there are many ways to build a tree. And, you know, if you get a kid, how do you make a tree out of cubes, like kind of Legos? Maybe Legos are more familiar to you. Mm-hmm. So, you know, the idea is like, you know, you see a lot of videos of somebody building a tree from Legos. Can you actually teach a machine to build that tree from Legos? So there's a lot of interesting work in the space of neural cellular automaton that are kind of learning the rules of building structure. So if I give you a final structure, can you tell me how it could have been built? And I'm very interested in this class of models because ideally, with a lot of these videos in light sheet microscopy, maybe we can do the same with cells. So we can ask, okay, if I see a time-lapse video of an embryo being built, can I come up with ideas of what were the rules that were used? You know, right now, we're very far from that, but there's very exciting incentive on kind of thinking about, okay, if I have a dynamic process, how do I represent it in a latent space? How do I predict from it? What classes of dynamical systems can I represent? Can I use them to represent branching, for example? You know, Lang is a branching phenomenon. So there's a lot of really cool space where, you know, you see particles moving through space and time, and you're asking what are the rules that lead to this movement. So it's a bit of an inverse learning, a lot of symbolic regression, but there's no clear tool for how to do that properly in a developmental context. We're trying to think about this from an origami point of view. So I am thinking quite a lot about inverse design, which means that, you know, if I have simple problems like a flat sheet and I want to fold it in a particular way, where should I add tension at the beginning to lead to this particular folding? These are questions I'm very interested in because This is kind of like, you want to ask, this is actually an image from a fly, right? Like you have these bands correspond to different layers of tensions, and those kind of guide how the fly is being folded and the organ is being folded. So it's a very interesting area. And in a more biomedical point of view, this is also very important because tension really guides how we're thinking about wound healing. So there's a very interesting kind of interplay between gene expression and tension that can guide whether we're healing faster and slower. And together with my collaborator, Dr. Yvonne Wapi from Columbia University, we are trying to understand what type of genomic interventions can we do in order to speed up how long it takes for a particular wound to heal in mouse context, but also hopefully in organoid models for human skin. So there's a lot of intersection coming from both the very basic science, the very biomedical science angle, and the main important thing to me is that we're in a space where we have a lot more opportunities. So, the previous talk, it was there was this sad thing of saying, okay, the data is there, but it's so hard to bridge the gap between the research and the clinical side. I think that's a real problem. What I'm very hopeful in the next few years is that by interacting more with our biologist colleagues, there's going to be much more opportunities for statisticians to be part of labs and kind of inform what type of data we collect and how do we intervene. So I'm very excited about this. I spent a lot of my time thinking about data analysis and hypothesis, but now I'm working very closely with my colleagues to test these hypotheses. And I think this is really where exciting biomedical research and basic science will happen in the next few years. I'd like to thank you all for your attention. I'm running a little bit over time and I apologize, but I hope you're very excited about all these cool things you learned. And yeah, I'm very grateful to CIFAR for their funding, and if you know anyone that would be interested in the problems that I told you, I'm always looking for postdocs and grad students, RAs. Yeah, so come work with us or email me to collaborate. Thank you so much.